I'm Fabian from ISB-CGC, and I'll walk you through our poster. As a quick background, ISB Cancer Gateway in the Cloud, ISB-CGC for short, is one of the 3 cancer cloud resources. We're funded by the NCI to assist researchers with accessing the Cancer Data Commons and help to drive adoption of cloud computing for bioinformatic research. There are petabytes of bioinformatic data from many large, multi-institutional research cancer initiatives. TCGA might be an example that you're familiar with. In an effort to improve reproducibility, these data from these programs have been reprocessed by the data standard services using a single set of analytics pipelines and hosted in what are called data commons. However, this amount of data, often stored in thousands of individual files, one per analysis output per aliquot, may be overwhelming to many. So we at the ISB-CGC have extracted these individual files, concatenated the data in them, added many relevant metadata fields such as sample type, cancer or normal tissue, case, sample, and aliquot barcodes, and hosted these tables in publicly accessible BigQuery ecosystem. We aim to generate these tables exhaustively for every data type in the genomic and the proteomic data commons. And we have already published hundreds of tables, including clinical, RNA sequencing, and protein abundance for a variety of programs. So what is BigQuery? BigQuery is an SQL-based data warehouse where tables can be browsed, previewed, just like an Excel file, and with only a small amount of knowledge of query language, researchers can quickly calculate summary statistics for any table in this wide array of data. It is also possible to join between distinct data types. So here I'm showing a query doing exactly that. You can join based on the case, case barcode or the aliquot barcode that I mentioned before, and this query is showing a join between RNA sequencing and protein expression stored in 2 separate tables, and you can obviously also include clinical data, join on all types of different data types, and then you can pull results from these joins directly into R or Python, where you can run further analyses on them, plug them into differential expression analyses or any type of R library that is available. And you can generate beautiful figures based on these data. And it's also possible to leverage BigQuery it has considerable scalable computational power. And the monetization model chosen by Google allows for data processing that is currently not feasible on a more traditional institutional high-performance compute cluster. In trials, we were able to calculate millions of statistical tests in minutes, and we were also able to calculate billions of correlations for the total cost of only about $1. BigQuery also has support for custom user-defined functions written either in Java or SQL that allows you to expand on the base functionality of SQL and wire more complicated analyses directly into SQL, where it can take advantage of that scalable compute power that I mentioned. So I'd also like to take this time to mention that we have $300 of cloud credits funded by the NCI for anybody that wants to start up and test analyses in the cloud. And we have a wide variety of statistical notebooks, as well as some of our own custom user-defined functions we have links for in our documentation at isb-cancer-genomics-cloud.readthedocs. We also have a lot of ongoing and past collaborations with research groups to leverage both the strengths of the cloud as well as our ecosystem to answer their research questions. So this first example is ongoing work with the Aquamenza lab, comparing a Black African-American breast cancer cohort to a similar white cohort to try to answer the question of why the Black cohort tends to have historically far worse prognosis, to try to identify some of the factors that might be underlying that. So using TCGA BigQuery tables, we compared gene, miRNA, copy number, and mutation data from those 2 cohorts. And then shown here is a microRNA gene expression network based on the expression enrichment of MIR200B, which we identified in the Black cohort. And if you're interested in this work, there's more data on Dr. Aquamenza's poster at this conference as well. The second collaboration is with the Shmulevich lab, where we tried to create a cloud-based framework to find synthetic lethal gene pairings in cancer. These synthetic lethal interactions, based on initial mutations unique to tumors, are promising targets for therapeutics with likely low side effects in healthy tissues. This work used multiple data sets stored in BigQuery, such as the TCGA pan-cancer and the CCLE data, and used them as inputs for the DAISY pipeline, which focused on pairwise gene co-expression, mutation pair absence in tumors, and CRISPR double knockout phenotypes to predict lethal pairings. Evidence from all this can then be combined to generate likelihoods for synthetic lethal partners for any gene of interest. And crucially, all of this work was performed in cloud-based notebooks and can thereby easily be replicated and modified by any researcher with a personal computer without a huge overhead of having to download the data sets and install all these pipelines on their local systems. So in summary, BigQuery is a powerful analysis tool that can scale analyses to an unprecedented level while also remaining inexpensive. The ISB-CGC, alongside the other cloud resources, hosts a vast amount of standardized cancer data that can be used for research right now. And we have many notebook resources available in our documentation for scientists with a wide range of programming skills, and we are continuing to expand the data available in our ecosystem. If you have any questions or would like to collaborate, or even just want to talk about whether cloud computing is a good fit for your lab, don't hesitate to contact us either at feedback@isb-cgc.org or at our office hours every Tuesday and Thursday, links for which you can find in our documentation. Thanks for your attention, and I hope you have a great conference.